One thing that's taking off quite a bit, especially in the areas of cancer research, is multiplexing. So a lot of people are using this for immunology applications, so trying to identify different immune cell types. So one kit which is used fairly regularly are the opal dyes. So in this case, again, we've got our antigen or our protein of interest. We have a primary antibody which is detecting that, and then a secondary antibody which is detecting our primary. Now, in this case, we don't have a fluorophore attached. We've got an enzyme called horseradish peroxidase, and what we're doing here is trying to set up a chemical reaction. So we have our fluorophore up here, which is an inactive tyramide fluorophore. When this undergoes the chemical reaction, which is set up by the HRP molecule, then it creates a reactive tyramide fluorophore. What this does is it actually binds covalently to the tissue, which allows us to basically set down the fluorophore directly onto the sample itself. What we can do then is come in with heat, often a microwave, or there are automatic systems that will do this now. And this will actually remove the antibodies from the sample, but the fluorescence sample will stay behind. Now, why do we do that? That's because we can then come back in with another molecule or another antibody to detect another molecule with a slightly different fluorophore, and lay that down on top. And we can do this multiple times, so we can actually get up to 7 or 8 antibody stains onto a sample, which we can image using a system which will spectrally unmix the signals. And as I say, this allows researchers to look at the different types of immune cells within a sample. The other nice thing is that means you don't have to worry about necessarily where the species that your antibodies were raised in, because the antibodies are no longer there when you come back in with a secondary antibody. Last thing in terms of the fluorescence molecules are quantum dots. So these are reasonably expensive, which means that don't tend to get used very often. They work slightly differently to traditional fluorescence, in that it's the size of the quantum dot that determines the fluorescent signal that's being released. Nice thing is that they don't suffer as much from photobleaching as a result. So these have a zinc sulfide shell, very small size cores, which is mixed with cadmium, selenium, or tellurium. We can functionalize the outside of the quantum dots, so we can attach oligos or antibodies to those to bind specifically to your targets of interest. And as I said, the emission wavelength is tunable depending on the size of the quantum dot. So these are some examples. You'll see they have essentially a very, very similar excitation profile, but have nice, relatively narrow emission peaks. So you could reasonably easily do a 4-color experiment here. In terms of probes and stains, again, this is a 1-hour lecture in itself. I'm not really going to go too deep into this, but there are a number of cell compartment specific stains, which a number of them are cell permeable, so can be used in live cell experiments. Again, I'm just using a tool that's on the Thermo Fisher webpage, where they're showing 3 of their different 3 different stains. So DAPI here is staining the nuclei that can be used in fixed samples. They've got a cell mask, which is specific for the plasma membrane. So this is in the deep red range. That can be used either live or fixed. There's a mitochondrial marker here. Again, can be used in a live. This is in the red wavelengths. And then there's an endoplasmic reticulum marker, which is in the green wavelengths, and again, can be used live. And again, there's an endless list of probes that are available. If you have specific compartments that you're interested in, then I suggest you do a little bit of research in terms of what's available to you.